Okay, so the next talk, I hope everything is going well. Yeah, so we don't have another session, so I'm going to. Next talk will be given by Emily Bao on real-time direct classification of nanopore signals with SquiggleNet. Joint work with, this will kill this extra minute, Jack Wadden, John Herb Downward, Piyush Ranjan, Wei-Chan Zhu, Torin McDonald, Ryan Mills, Alan Boyle, Robert Dixon, David Blau, and Joshua Welch. Okay, and now we are on time. Emily, over to you.
Thanks, Travis. Hello, everyone. My name is Yuwei Bao. I am a PhD candidate at the University of Michigan in the Department of Computer Science and Engineering. Today, I will be presenting our work on the real-time direct classification of nanopore signals with SquiggleNet. Oxford nanopore sequencers such as MinION or Promethean determine the nucleotide sequence of DNA or RNA molecule by measuring the changes in electrical current, which we also call squiggles, as molecule translocates through a protein nanopore. This approach is fundamentally different from the widely used Illumina platform and provides several benefits. The sequencer can streamline the sequencing in real time. It offers long reads, outputs, and the sequencers like MinION or Flango is really small, fast, and portable, making it ideal for rapid diagnostic and field work. The nanopore sequencers were then recognized in wide range of applications. For example, we used the MinION to sequence patient lung fluid sample in order to detect a pathogen genome for rapid real-time diagnosis. However, the problem was that we found out that over 99% of the genome in the patient lung fluid sample was actually human genome, which we don't care about. Our targeted genome, such as the bacterial or virus DNA, were far less than 1%. The MinION were therefore wasting most of the sequencing time and resources on those non-targeted reads. Therefore, the sample needs to be enriched. Here are some conventional enrichment methods, including PCR, CRISPR, hybrid method, and etc. However, they would usually consume a lot more time, equipment, expertise, and usually they would only offer limited results. There are also some alignment-based computational enrichment method. Some uses base caller plus sequence alignment algorithm. Some uses a rough base caller to match up with some k-mers. However, these alignment-based computational algorithms usually consume huge amount of computational resources, including GPU, huge clusters, and moreover, they would rely on a large reference database in real time when they're doing the computation. We then turn our eye back to our Oxford nanopore sequencers. Apparently, nanopore sequencer can eject a partially sequenced molecule, a capability referred to as read-until. The read-until API allows selective sequencing of molecules by reverting the voltage of each individually selected nanopores and ejecting the unwanted molecules. The read-until API has the potential to computationally enrich the sample DNA or RNA content, save the time and the resources for only the targeted genome that we're looking for. Therefore, we propose SquiggleNet. It is a fast, light, accurate deep learning-based classifier to computationally enrich targeted reads for nanopore sequencing with read-until API at the squiggle space, meaning bypassing the expensive base caller procedure. Here's the outline of today's talk. In terms of methods, the entire pipeline of the SquiggleNet is straightforward. The network is first trained to recognize certain classes of sequences, such as human versus bacterial DNA, using labeled examples. Then, as the nanopore sequencer generates raw electrical signals from the new and unseen sample and generates a bunch of squiggles, SquiggleNet then can rapidly classify each molecule to determine whether it is a sequence of interest or not. Molecules of not interest will invoke a read-until API to be ejected from the nanopore, freeing up the pore and for the sequence to run through another different sequence. In the contrast, targeted molecules are then accepted and sequenced to full length and used for any downstream analysis that you prefer. In terms of model architecture, SquiggleNet employs a convolutional architecture using a 1D ResNet-based model over squiggles. it only takes 3,000 signals, which corresponds roughly around 300 base pairs worth of input information, bypassing the expensive base calling procedure and directly makes an accurate and quick decision in the squiggle space. The architecture consists of 4 blocks with increasing number of channels, finishing up with mean polling and a fully connected layer with softmax for the final class prediction. We also experimented with several other approaches, including RNN, LSTM, GRU, combination of several of them, different hyperparameters, hierarchical, etc. However, we found that the approach based on the convolution actually outperform all of the other methods we experimented, suggesting that local features actually is sufficient for this problem. And also, usually those recurrent neural network-based models are hard to take a long time to train and hard to parallelize. We conducted a series of experiments to demonstrate the high performance of our model, the robustness to detect unknown species, the fast speed, and efficient memory usage, and the wide range of potential applications. To test our performance of the SquiggleNet, we generated 4 experimental data sets containing a mixture of human and bacterial DNA from the Zymo community. For the first data set, we achieved the ground truth labels through Minimap2 alignment. For the other 3 data sets, human, Zymo, barcode 1, 2, 3, 4, and 5, 6, we used the barcode to avoid systematic error from the alignment algorithms and obtained reliable ground truth labels, species labels this way. We discarded the first 1,000 to 2,000 signals of each read to remove the adapters and the barcode and prevent the model from cheating. The other 2 data sets were generated from the procedures linked in the paper. We split the first 2 data set into training, validation, testing, and used all the other data sets for testing. Noting that these data sets are from different sample preparation procedure, flow cells, and different composition ratio. So the first 2 are 1:1, the third was 1:4, human versus Zymo. These 2 are hugely disproportionate favoring human genome, and the last one is also 1:1. We also trained, all of the results shown later are using the first model, the model trained on the first data set. We also trained the model on the second data set, and received the similar results. Overall, our model classifies each molecule as bacterial or human with over 90% inaccuracy across different test data sets using only 3,000 signals per read. The recall and ARLC score are also well above 90% across different data sets. For the last 2 data sets, the precision scores were diluted by the extremely disproportionate human to bacteria ratio, but still exceeds the sample concentration by 10-fold. The classifier generalizes well to different lab preparations, flow cells, and the proportions to species. To further test the generalizability and practicality of SquiggleNet, we tested the model on the data set collected from several clinical human samples. This respiratory metagenome data set has a human to bacteria ratio of over 900:1, and the bacteria species overlaps with our training data set by less than 3%, meaning most of the bacteria species in data set were never seen during the training procedure. As shown in the previous figure, our model still achieves over 90% in accuracy and over 72% in recall, demonstrating the robustness to detecting these unknown species. We next compared the performance and efficiency of SquiggleNet against the previous state-of-art methods, Guppy plus Minimap2 and OnCalled. We benchmarked the runtime on the left and the accuracy on the right of these 3 methods with increasing read length from 300 base pairs to 600 base pairs to full length. The processing time of SquiggleNet with 300 base pairs of the input is among the lowest, and yet the accuracy is among the highest of all 3 methods. For the other 2 alignment-based methods, with longer input length, the processing time grows dramatically, whereas the accuracy gain is actually pretty limited. And also these 2 other alignment-based methods also relies on a 3.2 gigabase of a reference database. Moreover, SquiggleNet model itself is only around 300 kilobase pairs compared to the smallest Guppy model for the base caller is 5.5 megabytes. More specifically, SquiggleNet took around 300 seconds, which is about 13 minutes to finish processing on GPU. We also tested on the MacBook on CPU, and it took 2,600 seconds, which corresponds to around 43 minutes. We also observed that over 90% of the SquiggleNet processing time on the GPU and over 40% of the processing time on CPU is actually spent on loading the data from the disk, which can be significantly reduced while streaming line through the MinION in real time. Lastly, classifying species is useful for many applications, but distinguishing among different loci from the same species would actually significantly expand the settings in which SquiggleNet can be applied. To investigate whether SquiggleNet can be used to enrich loci of interest within the same species, we compared our method to a recently published enrichment protocol that enriched specific families of interspersed repeat elements using Cas9 directed adapter ligation. This experimental enrichment is effective, but still achieves less than 50% of the reads containing the repeats of the interest, whereas SquiggleNet easily achieved over 90% in accuracy using only 3,000 signals per read. And this experiment shows that SquiggleNet is not only good at distinguishing between human and bacterial reads, but also within the same species. And this just opened up a lot more application potentials. In conclusion, SquiggleNet is fast, it's highly accurate, it's memory efficient, it is robust to different sample environment, robust to detecting unknown species, and has a lot broader potential applications. For future application of SquiggleNet, it is highly recommended to retrain or at least to fine-tune on the specific data set that an experimental setting you're working with to achieve optimal results. As mentioned earlier, there's also larger room of optimization in streamlining the whole procedure to achieve even shorter runtime and save the time and the resources for the downstream analysis only on the targeted reads. Overall, this work could not have been done without the collaboration between Michigan Engineering and Michigan Medicine, and I'd like to thank all of my great mentors and the collaborators for their contributions. The open source model can be found in our GitHub page, and the training dataset has been uploaded to SRA. Thank you all for listening, and I'm happy to take any questions if there's any.
Okay, so now some questions have appeared. First question, is the method also safe for quantification of RNA? The method can maybe cause bias in the representation of targeted reads.
Is the method safe also for qualification of RNA? We assume it would, but we have not had the data set to experiment with, so we would highly recommend you experiment it with RNA. The method can maybe cause bias in the representation of targeted reads. Could you elaborate on what kind of bias you might be referring to?
I can't, I'm a data structures guy, but if the anonymous attendee would like to, in the meantime, we can go to the next question from Ben. Great talk, thank you. A possible advantage of your approach, since it accepts electrical signal, is that the network can observe base modifications. These might be good at distinguishing human from bacteria, e.g. adenine methylation probably indicates bacterial DNA. This could also be true for the repetitive element application, since repeats tend to have cytosine methylation. Do you know the degree to which the model is making decisions based on just ACGT sequence versus other information inherent in the squiggles, like base mods?
Yeah, that's a great question. We actually did look into whether our model is looking into, for example, DNA modifications such as methylation information on the signals, because other than those other methods, which does the base calling step first and just outputs AGCTs, the raw squiggles does include richer content that has those informations, but also potentially noisier. We found out that there is a significant difference between the methylation content between bacterial reads versus the human reads. In terms of the model interpretability, we experimented some very basic integrated gradient methods, and because the model is deep has a lot of layers, it is kind of hard to interpret exactly which layer is interpreting exactly which feature. So we want to believe that the model is definitely using all kinds of information directly fed from the squiggles. But in terms of exactly which feature, it's kind of hard to interpret. And I also do want to mention that we did the data pre-processing procedures to remove the first couple hundred base pairs because those are adapters or noise. We also did the across platform sample pre-processing, meaning we like divided by the standard deviation and subtract by the mean, just so that we can normalize all the data so that all the data is starting at the same place. But yeah, that is a great question. And we want to believe that our model is using all kinds of information and we just don't know exactly which one.
So we don't seem to have, if I know how to work this thing properly, we don't seem to have another question yet, or the follow-up to the first question.
Okay.
But I wanted to ask, so I think we have time. I wanted to ask, have you tried this in combination? So using SquiggleNet as sort of a filter and then running, say, Guppy and alignment-based methods afterwards. I know this sort of defeats the point of sort of saving space, because then you still need the database and all that kind of stuff. But if you could, I mean, there was one bar where you got 90% and somebody else who ran it on the full reads was getting 91. Could you like use SquiggleNet to throw the junk away and then just pass on the stuff that you're like, yeah, it could be what we're looking for?
Yeah, actually, the whole purpose of using SquiggleNet is not in replacement of Guppy plus alignment algorithm, because we are a binary classifier. Our main goal is to filter out all those non-targeted reads that you don't want. In terms of any downstream analysis, for example, you want further detailed classification down to species or even strand level, or you want to do variant calling or whatever you want, like you would probably still need to do Guppy plus whatever other methods you're using.
Oh yeah, but I was just saying that when the score comes out of SquiggleNet, is there some sort of like a quality score where it says, this one I'm absolutely sure is junk, this one I'm going to call it junk, but maybe it would be worth looking at some more?
Yeah, there is definitely that score. So the last layer out of the softmax, it's basically 2 class and a probability on both. So it's like, for example, one is 50%, the other one is also 50%. That means you're not super sure. But if one is 90%, the other one is 10%, then you're pretty confident it's going to belong to this one. So yeah, okay. We can definitely output that confidence score.
Okay. So sorry, that's I have a timer set. This means that you have 3 minutes left. So does anyone else have questions? Otherwise,
yeah, I've left our GitHub page and also maybe content info. Feel free to let me know if there's any follow-up questions or chat with me or future collaboration. I'm happy to chat more.
Okay, thank you. Yeah. Okay, in which, wait, there's, well, I still don't see any new questions. Okay, but maybe we should try to do a smooth transition. Thank you again.
Mm-hmm.